this video we will discuss about this PARP-14 was overexpressed in pancreatic cancer cells. So basically we will answer the questions why the researchers has selected PARP-14 and to study the impact of PARP-14 on pancreatic cancer cells. In the previous video we have discussed figure 2 and figure 3 where we have seen that PARP-14 promotes proliferation and chemoresistance in pancreatic cancer cells. But by mistake we have not discussed the figure 1, which basically give us the answer why the researchers have selected PARP-14 and its impact on the pancreatic cancer cells. So we will discuss that figure in this video. So what the researchers did is they analyzed the dataset that is publicly available of the pancreatic cancer patients, and in the 3 different dataset they are able to see that this expression of PARP1 is high compared to the normal tissues. Expression of PARP14 is high in the pancreatic cancer tissues compared to the normal tissues. In 3 dataset out of the publicly dataset available for the pancreatic cancer patients, they checked 3 dataset and they're able to see the similar trend. Thereafter they check the relative expression of PARP14 using qT-PCR or quantitative PCR, and there also they find the similar things, that is tumor tissues has high amount of PARP14 compared to the normal tissues. And once they are able to see the similar trends, they further validated these PARP14 expressions using Western blot, and that is they took the samples from the 4 different patients and analyzed the PARP14 expressions in the normal adjacent tissues and the pancreatic cancer tissues, and they're able to see the high expression of PARP14 in the tissue samples, pancreatic cancer tissues compared to the normal tissues. So this all experimental results, that is Western blot, quantitative PCR, and this this analysis of data publicly available dataset points out that PARP14 was overexpressed in the pancreatic cancer patients. And after that they also check the pancreatic cancer tissue slides using immune. They stain this pancreatic cancer tissue slides and the adjacent tissue slides, and there also they see the similar trends, that is pancreatic cancer tissues has more expression of this PARP14 compared to the adjacent tissues, that can be seen clearly. And finally they check the cumulative survival analysis of the pancreatic cancer patients that has high expression of this PARP14 compared to the case where PARP14 expressions are lower, and they are able to see that patients that have high expressions of this PARP14 has low survival rate compared to the case where PARP14 expressions are lower. So this final cumulative survival analysis indicates that this PARP14 is directly related to the survival rate. That is, when the PARP14 expressions is high, the survival rates are low, and when the PARP14 expressions are low, the survival rates are high. So this figure in all concludes or made a point that the PARP14 expressions might be helping the pancreatic cancer cells to progress more aggressively, and that is the reasons the patients that have high expression of this PARP14 dies earlier compared to the case where patients that have low expression of this PARP14 die slow. So this is the basis for their further research, that to study the impact of PARP14 on the pancreatic cancer cells. And In the figure 2 and figure 3, what they have shown is that the PARP-14 promotes proliferation and chemoresistance in pancreatic cancer cells that we have discussed in the previous video. So I hope you understood the reasons why they have selected PARP-14 and its impact on the pancreatic cancer patients. And before even doing this analysis, the literature also shows that PARP-14 has some tumorigenic properties and helps and is found in most of the cancer patients, but its impact on the pancreatic cancer patients has not been studied. So that is the reasons they started the analysis from this publicly pancreatic cancer data available thereafter qT-PCR and Western blot and uh, immunostaining of the pancreatic cancer slides and adjacent slides, and this overall survivals of the patients and its relationship with this PARP-14. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.